cellular respiration. All right, so remember, this is going to be how all living things extract energy from the molecules that they either create in the case of photosynthetic plants and the rare photosynthetic organism, or more likely they harvest as just about all heterotrophs. So the common terms you got to keep in mind are ATP, adenosine triphosphate, our energy molecule, what the term electron acceptor means. And remember, that's that temporary shuttle just to carry the extra energy and to move it from point A to point B. And then ADP, adenosine diphosphate, which is going to be the same thing as ATP, but now it's missing that last high energy phosphate group. And so this is the cycle we're going to see. ATP will break, that energy will be picked up by an electron acceptor, it will be utilized somewhere, and then we're going to have a process to recharge the ADP, turning it back into ATP. So we'll start with the basics, beginning with glycolysis. So glyco, sugar, lysis means break. Remember, this can happen anywhere, anytime, basically. If you have oxygen, that's nice, but it doesn't need it. So for a lot of organisms, prokaryotic, bacteria especially, you're going to be looking at glycolysis being their only source of energy. They don't have all of the advanced organelles. Many of them have found ways to live in anaerobic environments in the muck at the bottom of rivers and swamps and things like that. And so, this is very important for them. And what we're going to do is we're going to take that glucose sugar and we're going to break it down. Now, it would be great if it was as simple as this is, but it's actually a good deal more complex, and this should look pretty familiar to you guys. So, glycolysis is going to involve several steps of breakdown and reconfiguring. Throughout the series of doing so, we've got to start it with 2 ATP. We need the energy to start the ball rolling, and from there, everything will kind of cascade on its own, but it does take that initial investment. It's going to make for us a little bit of electron acceptors right here. And in the end, it's going to produce 4 ATP, but since it took 2 to start it, we really only count it as a net gain of 2. Now, this is the entire thing fully broken down with the little dots for each of the carbon molecules so you can see step by step how those have been changed. What this is when we take this fructose and we break it, half of them go over here and go through this series. The other half ends up over here, and it's really the same thing, but they've got this one intermediary here due to uneven cleaving of electrons and charges that needs to go through a little bit of adjustment before it can follow the same pathway as the more friendly start there. Now, for cellular respiration, this is the important part because glycolysis happens out here in the cytoplasm, but for anything else to occur, it's got to get here into the mitochondria using this intermembrane space right here between the inner and the outer membrane, and then the matrix and the cristae of the inner membrane. Now, in order for the pyruvate to get there, it does need to get slightly changed. Basically, all we're doing is we're stripping it of a couple of electrons, generating some more electron energy acceptors. And this is where the CO2 waste gets kicked out. Then it enters the Kreb cycle, and you don't need to go through every step. You just need to understand what it is, and it is the taking of the acetyl-CoA and then cranking this through twice, because technically you're going to get 2 acetyl-CoAs, because at the end of glycolysis, we're ending with 2 pyruvates. So each one of those pyruvates will go through the intermediary step, get converted from pyruvate over here to acetyl-CoA over there. And that this will make a couple ATP. The important thing, though, is going to be the products down here, the energy acceptors. 6 NADH, 2 FADH2s, meaning that we are coming out of this with 8 electron acceptors, and that's going to do some real work for us. Got our key terms. Hopefully you guys remember them. This is the raw materials that come out at the end. They go off. And then the electron acceptors that are going to stick around and move on to our next stage. And that is the electron transport chain. Now remember, glycolysis happened out here in the cytoplasm. Krebs cycle and electron transport chain have to happen inside the mitochondria. And this is the simplified view of the entire thing. And I know it still looks complex, but this is still simplified, right? We've got our little inset of glycolysis, Krebs cycle, and etc. And then this is looking at just a particular section of the inner membrane. And it's identifying where is the matrix and where is the intermembrane space. Notice that intermembrane space is the area that's going to be filling with those hydrogen ions, those H pluses. All of our reactions that are happening inside the matrix are going to be generating the protons. The protons are then going to get pumped out of the matrix into the intermembrane space, creating a chemoosmotic gradient where we have a high concentration of them outside, a low concentration of them inside, and so naturally they want to diffuse back into the matrix. But the only way to do that is to go through the ATP synthase. This enzyme over here, we know it's an enzyme, ASE at the end. And as those hydrogen ions pass through, that will oxidatively phosphorylate our ADP. It will reconnect with the missing phosphate group that was broken off earlier in glycolysis or just laying around from other biological processes, regenerating our ATP, and we are ready to go for some more. Now, when we went over it, I compared it to photosynthesis. Because where photosystem 2 is going to be generating a lot of those H plus ions inside the thylakoid, remember that here we're going to be doing something kind of similar, but now we're going to be generating them and then pushing them out to the actual inner membrane space for the H plus ions. In total, glycolysis makes 2, Krebs cycle 2, electron transport chain. This number can vary depending on how efficient it is in moving its energy, but anywhere from usually about 28 to 34 is accepted, with 32 being the average that it generates, and therefore 36 ATP on average being the accepted amount that we get from a single glucose molecule. During cell respiration, and I had written this terrible before, so here's a much nicer version of that reaction. Understand that a lot of the energy that does get released is going to just plain be lost. And it's going to be lost as these wavy lines that emanate out from the organism doing it. It's going to be lost as heat. We talked about that with Gibbs free energy and, the different laws of physics. Now, oxidative phosphorylation, I wanted to explain a little bit more because last time we really kind of glossed over it. So here you see the hydrogen ions coming in through the ATP synthase. Now remember, that's just a proton. It has a positive charge because it's a proton with no electron. So the positive charge of the proton overpowers the atom entirely, and so it's a positive charge. What's going to happen is as this comes through, and the ADP with that phosphate are brought in, this H plus is going to actively kind of pull electrons away from our ADP and our phosphate. It's going to be relieving them of that only for a moment, just long enough for each of them to realize that they need something else and then for them to bond together as our ATP. Oxidative phosphorylation means that we are going to remove electrons to add a phosphate. Be careful, the term oxidative, this is pure chemistry. This is not really biology related. So we're just talking about losing electrons. We're not talking about needing or functioning with oxygen at all. It does need it. You can see it over here. but it's not involved in the oxidative phosphorylation itself. And then we had to finish up talking about fermentation. Because for those poor bacteria that can't go on the Krebs cycle or electron transport chain, the problem is the breakdown of glucose to pyruvate happens through glycolysis, but now they're stuck with these two electron acceptors. They need some way to lose that extra energy so that it can be regenerated back to its hungry form so that it can pick up more energy again. And that's all fermentation is. Fermentation is just a way for bacteria, for prokaryotic cells to regenerate their electron acceptors to get them back to the point where they're usable. Otherwise, the entire process would just grind to a halt. And one of the examples that we can see of stuff like that is what we call oxygen debt. It happens to human beings when we exercise beyond our normal, excuse me, beyond our normal amount. And basically, it just means that your body, your muscles are burning glucose like normal, but at some point, we cannot keep up with the oxygen demand for the amount of activity those muscles need to maintain. The ATP is running low. And so, in an attempt to provide more ATP, the body kind of goes into anaerobic respiration, just glycolysis. And the entire time it's doing that, you are building up lactic acid because we don't have the rest of cellular respiration. We only have this. So lactic acid has to be fermented in order to regenerate these electron acceptors in order to continue doing this to provide more energy because you're running a marathon, you're 45 minutes into what's going to be a 3-hour run, and your body just knows we've got to keep going, but we're just not getting enough oxygen. Start making the lactic acid. We'll clean it up later. And then that's the cleanup process. As you breathe in more oxygen, it allows us to basically break it down, convert it back over into harmless things. Boom. All right. Do you guys have any questions regarding the basics of cellular respiration? Now, if you take a look at the worksheet, once again, going through here, it kind of reviews it. This is their unbelievably bad mitochondria. But it's got all the different steps here. It's showing what they're making, where they're going. It's really not a bad guide. But what I want you guys to pay attention to in particular is if you go down through all of this. Now, when we did it, we didn't really look at these extension questions. We were just trying to get the basics. Now, you guys should be able to do a better job in many cases of going through these if you did not before. So, I just talked about number 19. I want you guys to make sure that you take a look through them and try to answer them. Try to go through and figure out exactly what would be there. Tomorrow we'll sit down and we'll actually talk about it, go over each one, and then we'll start working into photosynthesis. I am interested in doing something later this week. Hold on, I'll go into that in a moment. I'm interested in doing a flipped lesson towards the end of this week, probably Thursday or Friday. So for whichever day I get everything together for, what will happen is inside the folder for the day, there will be another folder that will say flipped. And that's the stuff that I need you guys to do before you see me the next day. So basically, it's going to be like slides or a video or something for you guys to take a look at, to take your notes or whatever. And then when I see you, I want us to be able to actually try to work through something and be able to explain it step by step. All right, so with that said, we have our basic plan for the remainder of the week.